So it's my great pleasure to close out the session with introducing Hopi Hoekstra. Hopi is a professor at Harvard and also the curator of mammals in the Museum of Comparative Zoology. She's completed fundamental work on improving our understanding of the genetic basis of morphological and behavioral traits that affect the fitness of organisms in the wild. I would take up a whole speaker time if I tried to enlighten you with all of her amazing accomplishments. Suffice it to say that she may be one of the youngest members ever elected to the National Academy of Sciences. So I'm going to finish off by just reading the last paragraph of Hopi's bio for this course, and it illustrates just how wonderfully intertwined the Grinnellian legacy is. So Hopi completed IB 104 during her senior year at Berkeley. She did the Natural History of Vertebrates course, fell in love with field work and headed off to grad school and was mentored by Scott Edwards, who TA'd IB 104. She then completed a postdoc with Michael Nuffman, who took IB 104 and is now our present director, and is now at Harvard in the museum where several of her colleagues also took IB 104. Welcome.
So before I begin, I wanted to just share three things that sort of have come up in talks before. The first one, I was struck by these stories of rejection from the NCC. And so I wanted to commiserate with you, David, because I too have a story of rejection. And that is when I was a postdoc, my dream job came open. It was to be a curator at Berkeley. And I came and I interviewed, and I don't think I've ever been so nervous because I was interviewing in front of all my former professors and so forth. And I had a great time, but unfortunately I didn't get the job. They hired Rory. Okay, not a bad choice, but still my job. The second thing I wanted to say was I wanted to apologize to Hannah, here, for not taking you as a grad student. Well, I mean, it was nice to see all the great work that you're doing now and how your students at Archer Academy are benefiting from all your enthusiasm. So it all worked out. And then the third thing I wanted to do was make a confession. And so a lot of the talks this morning really emphasized how 104, as I knew it, has influenced the teaching, and that's being passed on now in courses at different universities. So at Harvard in the MCZ, it's so confusing, we have a rich history in teaching meology. So Scott Edwards teaches ornithology, he's here. Jim Henkin teaches herpetology, he's here. But mammalogy hadn't been taught for years, and I think the hope was that I was going to get there and teach mammalogy. But I don't teach mammalogy, and I feel very ashamed of this. But what's even worse is that now it is being taught by a herpetologist. But Jonathan Losas has taken up the reins and is teaching mammalogy, which, yeah, someday we'll correct that. Okay, and that also sort of explains why today what I want to do is tell you about the influence of 104, not on my teaching, but on my research. I thought I was so clever pulling out my notebook. I pulled out my notebooks and my class notes, and these have traveled with me all through my career. I don't have any other class notes but these notes. And so I took the course as an undergraduate in 1994. I had what I considered the dream team of instructors. We had Jim Patton on mammals, Harry Green on herps, Ned Johnson on birds. I'm sure we can all argue about what the best teaching was, but I felt incredibly lucky. And as a lot of us have expressed, it's really the instructors that made the course, right? And so I'm very grateful to all of them. So this class really taught me the love of the natural world. It taught me to explore, to wonder, to be curious. It also taught me incredibly great penmanship, like who writes like that? And I almost let the slide out because I was scared that Greg the lawyer was going to be jealous of my grade. and Michael too. Yeah, so I was one of those, I think, overly eager students. So I'm very thankful to Harry and Jim for putting up with me, because I'm sure I was in the museum hanging around more than they actually wanted me to be. And it also taught me something about discipline. I still can't believe, especially looking at college kids today, that I was able to get up for an 8 a.m. Field trip every Saturday for 3.5 months, especially because, I think I can say this here, I was in a sorority, so I had things to do on Friday night. And during this course, I also made incredible friends. So I came to Berkeley not because of its prestigious biology program, but because I got to play Pac-10 volleyball. And so just as photographic evidence, this is me. We were rated by height. I was the shortest on the team. But I took IB 104 with Tasha Kamenat, who if not, isn't here yet. Right here. So Tasha is a lifelong dear friend, and I should note, she's 5'10", I think, and she's still in the front row. So together, and I knew her from volleyball, but it was really bonding through this course, and I think the two of us probably made thousands of note cards, flashcards, to learn all these natural history facts and concepts, and we sat in the back of the team bus talking about biology. And we took the Friday afternoon lab because that was the one time it didn't conflict with volleyball practice. And we also had the best teaching assistant, Liz Hadley, who was also a lifelong friend and collaborator. And then there was Kevin Burns was the head TA, but he was scary because he was the one who had to enforce all the rules. Okay. So I love the lectures and the lab and the camaraderie and the people while taking the course, of course, but at that time I had no idea, no concept that this was going to have such a long-lasting effect on me as a person and me as a scientist. So 104 really taught me this appreciation of natural history, not only sort of real-time observations, but that rich history and literature that goes back hundreds of years that I'll tell you I've taken advantage of over time. And 104, more indirectly, as Rory sort of mentioned, had this huge impact on me because all of my former mentors are somehow involved in the course. So you left out only Jim Patton, who was an undergraduate mentor of mine. I labeled some mouse bones in my days. Then Scott Edwards is my PhD advisor, and Michael Nachman. And I just want to say, you guys remember when you were grad students and you had to give a talk in front of your advisor? I had to give a talk in front of all 3 of my advisors. So nervous. So you'll see the imprint of their mentorship on the research I'm going to tell you about today. So for me, when I became an independent PI, this was the natural historian who had the most profound impact on my research. He did spend some time at Berkeley at the very end of his career, and many of the specimens are cataloged in the museum as well. But he was born in Connecticut. This is Francis Bertodi Sumner. But he really was raised in the Oakland Hills, and he spent his sort of formative youth essentially doing what we did on 104 field trips, that is exploring the local habitat. And he didn't go to school till he was 10, which I think sounds pretty good, and was taught by his father, but spent most of his free time out there catching snakes and lizards and etc., and becoming a zoologist and natural historian. Well, he went off to Columbia to get his PhD, but spent his summer in Woods Hole, where he was continuing to do research, but also in part because he was courting a lady who was at Woods Hole in the summer. But this isn't relevant because Woods Hole was a place where he also had some more time to explore the natural world. So out in Cape Cod, he did his first collecting of Peromyscus, the mice that will make up the bulk of his research. So there, I like to think he fell in love not only with his soon-to-be wife, but also with Peromyscus. So he went on to Scripps Institute, before it was an Institute of Oceanography, and there really dedicated a lot of his career to driving around the US, collecting mice, and documenting their natural variation. And this is just, you can see in his stories, in his papers, this is a painstaking work, took a lot of time, many, many years. But as you read through these papers, in addition to all of the data, you can see that these natural history studies really raised some, I think, fundamental questions for him. And a repeated theme through these stories are asking these questions of, in this variation that I'm documenting, why did these traits evolve? Was it natural selection that was driving this, or other forces? So this is sort of what I think of as evolutionary mechanisms. And then second, how did these traits evolve? And what's especially interesting is that when he was doing these studies in the early 1900s, Lamarckianism still abounded. So he still wondered, are these differences caused by Mendelian genes? And if so, what are those genes? And these are really the two fundamental questions that have laid the foundation for the research that I pursued over the last, oh my gosh, 2 decades or so. It's really frightening to say out loud. Okay, so depending on who you talk to, there are about 55 species of Peromyscus. Some are really fell in love with one, maybe two, but this is one of them. And I think for very good reasons. So let me introduce you to Peromyscus pallianotus. You do not trap these in 104 because they're found in the southeastern US, so Georgia, Alabama, northern Florida, although closely related to Manipulatus. And they're called the old field mice because they occupy these overgrown agricultural fields. These are 2 of our field sites close up. Here's a less glamorous field site. This is a burnt peanut field in Alabama. More glamorous field site on your right is one of the beautiful white sand beaches of the Gulf Coast of Florida. And one of the reasons that he fell in love with this species is because they were so variable across this range. So here he is in his field gear. Yeah, for a little sunshade, turn to the side. And as I mentioned, he went out and just caught hundreds and thousands of Peromyscus. And one of the first things he noticed, you don't need to read this, this is one of the papers he published on the tremendous amount of color variation that occurs across populations within the species across the range. This was published in the 1920s. And as I mentioned, many of these specimens are actually in the mammal department here, and I've come and visited and used those specimens, looked at their coats, and then get little snippets of their skin for DNA as well. He really focused on the extreme variation and was really preoccupied for many years with differences like this. And one of the things he noticed, perhaps not surprisingly, is that their coats really closely match the local substrate in which they inhabited. And in those papers, he wondered about what was driving these color differences and what the underlying genetic mechanisms may be, once he convinced himself that they were indeed genetically encoded. So I'm not going to go into detail. I'm sure Hannah could teach this better than I could. But we did experiments. So this was an experiment done by Sasha Vanieri, who you heard from earlier when she was a postdoc in the lab, where she made hundreds, if not thousands, of little plasticine mice, painted them either dark or light to match these natural populations, put them out in dark and light habitat, and luckily fooled the predators who would come and attack these models, and was able to show quite convincingly that being a mouse that matched your local substrate, like these top 2 images, you were attacked less often by visually hunting predators than those that were mismatched. And so it was a nice demonstration of the role of natural selection in driving these changes, or at least contributing to these changes. And then using more modern molecular tools, we were able to hone in on the genes that contribute to these differences, one of which is the melanocortin-1 receptor, which Michael made famous in his studies of pocket mice. And in these guys, we were able to identify a single amino acid change, an arginine to cysteine change of this receptor that we learned affected the way the agonist binds, the way the protein folded, the way it signaled, which eventually led to the light coloration between these. So in doing so, what we did was capitalized on all this beautiful natural history work that Sumner did nearly a century before but using modern molecular techniques, addressed questions that he wondered about all the way back then. And along the way, we learned some really cool things as well. For example, the mice on the Gulf Coast of Florida, these beach mice, which are lighter in color than the mainland mice, had this mutation in the melanocortin-1 receptor that made them lighter. But mice on the Atlantic coast that had independently evolved very similar light coats didn't have that same mutation, and in fact didn't have any new mutations in that gene that made them lighter, suggesting that even within the single species that there were different genetic solutions to this common ecological problem of wanting to be a light mouse in light soil. And we also learned that sometimes highly divergent mammals would use similar genetic mechanisms. So I usually ask this question to a less well-educated audience, so I'm just curious what your answers would be. So what I would often say is, what is the, if you just had to guess, what's the most divergent mammal from a mouse? So what would you, all of you, you've been educated by 104, the best of the best, what would you say?
Platypus.
Platypus, right, which is not the answer I'm looking for, and not the answer I ever get when I give this talk. So most often the two answers I get are whale or elephant. And I'm usually going for elephant. You might just know too much, I knew that would work. Because it turns out that when folks were extracting ancient DNA from mammoth bones, they sequenced the melanocortin-1 receptor and found a mutation in that receptor. It turns out it's the exact same arginine to cysteine change with the homologous position 65 that suggests then that mammoths, like beach mice, may have been polymorphic in color as well. So here, highly divergent, maybe not the most divergent mammals, but highly divergent mammals use the same genetics. So while working on these mice, however, Sumner made another really important observation with his then graduate student, Carol. So I'd love to show this picture, in part because we all know we sort of imprint on our graduate advisors. I like that nicely. So while the coat color of these mice is highly variable, that I didn't see coming. The coat color is highly variable in these mice. One thing that's not variable is the way they build burrows. And in this sort of, it's like a natural history note in 1929, they published this paper that basically had gone forgotten. And they described the unique burrows that these mice make. So they're characterized by an entrance, a long entrance tunnel, a nest chamber, and then a secondary tunnel that comes up towards the surface, but importantly doesn't penetrate the surface. And you can find these in the field because if it's an active burrow, they'll have this plume of freshly excavated soil out here. Sometimes they're plugged as well. So next what I want to do is just tell you about how we catch these mice in the field because it's going to illustrate two important points. Okay, so the first thing is we go out to the field. So this is like the beaches of Florida. We usually go in February. Remember, I live in Cambridge. So we go out and we get lots of volunteers want to come down, and you find an overgrown agricultural field, and you look for things like this. So this is this hole. Here's that plume of freshly excavated soil. And then the next thing you do is you get a graduate student. You get that graduate student to lie down on their belly, this is Jesse, face in that burrow. And while it looks like we're standing around laughing at him, we have an important job to do. So what Jesse is doing is he's threading tubing down that entrance tunnel until he hits the nest chamber. And he's going to blow in that tubing and sort of agitating anything that's inside, presumably mice. And we're standing around because what's going to happen is those mice are going to come flying out of that secondary tunnel, which we can't see, and we jump on them. Who needs a Sherman trap? And this is actually really effective. And I tell you this because there's two things that we learned from this. The first is that secondary tunnel is really used as an escape hatch, such that if something comes in the entrance tunnel, like tubing or maybe a snake, they have a way out of that. The second thing we learned is that even though we can't see where that secondary tunnel is coming out, it doesn't penetrate the surface, the burrows are so stereotyped in their size and shape we can predict. And that's the only way that somebody like me can catch these incredibly fast mice. So what we're doing now, I'm not going to show you any hard data, but what I wanted to tell you now is that based on this natural history observation, we're trying to understand why this complex burrow structure evolved and how differences in burrowing may affect fitness. So we're starting with some very descriptive natural history stories. So here are a student and a postdoc from my lab who've gone out to Florida in February. They're trapping all the mice in a particular grid. They each get a little RFID tag. Then we've built antennae that we can put around the entrance of the burrow that allow us to document every time a mouse goes in or out of that burrow. And in doing so, I hope this works. Sorry, this is not National Geographic quality video, but cute little mouse. So we're starting to learn all these things about the lives of these mice that we had no idea. So in each one of these traps, we have a triggered camera and a video camera that are run off of car batteries. And we can watch them, how much time they spend burrowing, who goes in, who goes out. We've always thought about one burrow, maybe a pair of these mice. Turns out they're highly social. They're running into each other's burrows all the time. They're big hordes of mice galloping across the landscape together. Things we never would have predicted. So in this study, we're also trying to understand more about the underlying evolutionary mechanisms. And we're also going after the genetic mechanism. So we were able to bring these mice from the field into the lab. We built these big boxes, filled them with 1.5 tons of dirt. And in each of these boxes, we put a mouse. We can leave it there for 2 days. We let it do its thing, and we can measure their burrows in a highly sophisticated way using this stuff. It's the kind of stuff you would buy at Home Depot. If you have a hole in your wall, you squirt it in, and it expands and fills that hole and hardens. So what we do is we make casts of their burrow, and it's a way of measuring the behavior they've done over those 2 days and transforming that into a very easily measurable morphological trait. So here's what we do in the lab, just like the field. There's that tube going down. Out. I have no idea where they're coming out. And then we fill that same tunnel, making sure that the mouse is taken out of the box. Very important. We fill it with what we refer to as our phena foam. And this phena foam is you see it coming out the other end. You leave it in there, it hardens, and then this is where having lots of eager undergrads comes in handy. They have to dig these out, right? And this results, so each mouse we run lots of times and results in undergrads are scaled. This is early days. This is why it's nice that we have an attic in the MCZ. Fill it up with this stuff. When people talk about big data, I always have to chuckle. And then we collaborate with folks in applied math so we don't measure these with rulers, but we use computer programs that can identify the nest, measure the curvature, the volume, et cetera, et cetera. And we've really focused on things like burrow length. So there's a small burrow and a long burrow, and the presence or absence of this escape tunnel. So I'm just going to tell you very briefly a little bit about the genetics. And that is, Polynotus builds these complex burrows, but its sister species, Maniculatus, digs these relatively dinky, almost sock-shaped burrows. And we could take advantage of the fact that these two closely related species, well, good biological species, if you bring them into the lab and don't give them any choice, happily mate, breed, and produce viable and fertile offspring. And so what we can do is cross these two together, make an F1 hybrid, and then ask, what does that F1 hybrid do? And we were making bets in the lab, was it going to build a big burrow, small burrow? And we were actually quite surprised to see that they in fact make burrows that are completely complex in the sense that they're statistically indistinguishable in size and shape from Polynotus. So here's just some of the data. The entrance tunnel is just as long as Polynotus, and they've always built escape tunnels just like Polynotus does, but Maniculatus doesn't. So to us, that suggests that there's some dominance involved, and it informed the next step of the experiment, where we could take the hybrids, breed them with Maniculatus, and then ask what do those offspring do. So using this breeding approach, we're able to localize regions of the genome that contain genes or genes that ultimately contribute to those differences in the innate component of those burrow building behaviors. And we were able to identify 3 regions that are shown in black on different linkage groups or chromosomes associated with the length of the entrance tunnel, and a separate one associated with the escape tunnel presence. But it turned out that we realized that if we now are narrowing down regions of chromosomes and we wanted to find the actual genes, like we found pigmentation genes, that if we were going to genetically modify these mice or so forth, the size and length of a burrow is actually a pretty blunt tool. Like, could we tell differences in 1 centimeter here or there? It's going to be hard. So we realized that we had to actually measure the mouse burrowing itself. And we were really interested in what are mice doing differently that give rise to these differences in burrow shape. But here's where we sort of started by scratching our heads, because this is a behavior that occurs underground and at night. So how are we going to measure this behavior? We got really lucky because I had this fantastic postdoc who decided to build the mouse equivalent of an ant farm. So this is a 2-dimensional plexiglass sort of structure we could fill with sand. The mice would burrow, and then we could see the burrows they make, and we could watch them doing it by just videoing them. And in doing so, we learned all sorts of neat things, not only like how totally cute these mice are, but like they turn upside down to dig out their nest chamber. They repeatedly go back and forth and up and down. And so we were so excited we could measure mouse burrowing, and then we realized, but how do we quantify this? So each burrowing episode was 8 hours. We measure each mouse 3 times. You know, even eager Harvard undergraduates, like Berkeley undergraduates, right? That was too much video. And then what do you measure? Like how many paw strokes they do, how smoothly they do it. It was a nightmare. So we went from being super excited to, oh, another challenge. But this postdoc said, well, here's what we'll do. Let's just automate it. So here what you're looking at is a mouse burrowing. This is Polyanotis. 8 hours sped up 900 times. The white blob is the mouse. And what we keep track of is 2 things. Below is the visible burrows that you see, and on top is the excavated soil that's coming out. And those 2 things are correlated. And so now we could watch and measure what the mouse was doing, when it was doing it, how fast it was doing it, but all in an automated fashion, really quickly. This got even more interesting when we compared it to Maniculatus, because here's the same assay. Maniculatus is very active, runs around, even builds itself this little ramp here. Every once in a while. But for the first half of this assay, it's doing anything but burrowing. Run around. Sometimes it takes a little break here and there, but runs around. And then all of a sudden, it's like something clicks. It realizes the lights are going to come on, and maybe I should make a quick little burrow, and it does so. This guy builds one here, and he builds one there, and then basically he sits in this burrow for the rest of the life. So using this burrowing, sort of automated burrowing analysis, we could learn all sorts of things about what the mice were doing, and it's given us now an opportunity to start to probe those genes we think are involved and ask how it affects mouse behavior. So I'm not going to go into detail. I do realize this is a natural history sort of celebration, but we're using things like genetics and pharmacology and viral vectors and chemogenetics, and we're even making transgenic paramyscus. That's a blue testicle, in case you were wondering. The point I wanted to make by telling you all of this is that this research program, which has occupied me for the last 10 years, is really all stems from this single natural history paper published in 1929 by a natural historian and his graduate student. And it was the questions he raised in those papers and others about evolutionary mechanisms and genetic mechanisms that were really the inspiration. So I wanted to end by highlighting something that I think we all know. I feel like I'm preaching to the choir a bit here, is that these natural history studies and observations can form those very interesting questions that we can dig deep, whether you go mechanistic in terms of thinking about evolution or thinking about molecular biology. Those can be really important observations that either you make yourself or your students make that formulate new projects and questions, or you can be slightly more devious like me and then steal those natural history observations and questions from others and address those in different ways. And importantly, the second point I wanted to make was that those natural history observations that we're making can have sort of real-time implications, or 100 years later can inspire a whole research program. So I'm sorry Ray Huey didn't make it, because he was the person, so again in breeding, right? So Ray Huey, who was involved in 104, was a committee member when I was a graduate student. And at some point during my graduate career, he said something like this. So it's not a quote, it's sort of a sentiment. Sometime in the late 90s he said, was that many great observations have been made and questions have been asked, but with new knowledge and tools and approaches, we can answer them in once unimaginable ways. And it's really this sort of connection between natural history and modern molecular tools that I think has been so important to me. And all of that appreciation of natural history, the natural history literature, all comes back to 104. So with that, I just want to leave this up there, because looking at my notebook, I realized that March 18th of 1994 was the first time I probably saw a Parameskius. And again, at the time, I had no idea that this stinky, bitey, jumpy, hantavirus carrying Lyme vector was going to become the love of my scientific life. So thank you.